This is video for Unit 5.2. We're going to continue our discussion of gene regulation in this video. And for this one, we're going to look specifically at eukaryotic cells, or cells with a nucleus like all of your cells have. So the first level of regulation in a eukaryotic cell is how the DNA is packaged. So you have over 6 feet of DNA in each one of your cells. That's a staggering amount of DNA to fit into a little tiny cell. And part of the way it fits into there is it gets folded and wrapped up and bunched up in a specific pattern. You'll see in this picture right here, oops, get my pen. Okay, this picture right here, we have these little like spiky things. They look like landmines. Okay, that the blue DNA is wrapped around. That's one of the first levels of DNA packaging. Each one of those little spiky balls, so they look kind of like that. Okay, single purple one. That is something called a histone. And together, the whole big clump of those spiky balls is called a nucleosome. And what you see on the right here are two pictures of what the DNA actually looks like under a really powerful microscope. So the first one at the top is not packaged into the nucleosomes yet, and the second one on the bottom is, and it looks like kind of beads on a necklace. So you can see the little nucleosomes. So it actually is visible with a microscope when DNA is bundled up this way. So again, this helps the DNA to get all packaged up. Now, the interesting thing about these nucleosomes and histones is they don't just help to package the DNA in the cell. They are now finding out that these things help to regulate the DNA as well. And this is part of what that movie Ghost in Your Genes talked about. This gets into the epigenetics. So let's look at this a little bit more closely. Remember, this histone, okay, is what we're going to be focusing on, and it's a little protein molecule with this little flag sticking out of it. Okay, so if we take our histones and we methylate them, and by methylation what we mean is we're adding a group to it called a methyl group, so CH3, a carbon and 3 hydrogens. The more histones we methylate, so again, here's our little histone, and we put a little CH3 tag on it. So the more histones we methylate, the more coiling that DNA is going to do. So if that DNA is coiled up tighter, that means there's less access to the genes, right? Because the DNA is all coiled up tight, so it's hard to get in there and get at the genes to transcribe them. The opposite of this is if we have more acetylation, which means less methylation. Again, acetyl is just another chemical compound that goes on there. So if we have more acetylation of our histones, that equals less coiling. Did it again. Less coiling, which means the DNA is stretched out a little bit more, and so there's more access to the genes. So essentially, methylation is like turning some genes off, and acetylation is kind of like turning some genes on. More access or less access. So this is a way we can regulate or change our genes. Now remember, as I said in the movie, methylation can occur from the environment. So things that happen to you during your lifetime can create more or less methylation on your histones of your DNA. And this again can be passed on to your offspring. So you are regulating your genes with your environment. Now finally, the last thing I want to point out on this topic excuse me, is just to kind of wrap up what our definition of epigenetics is. So epigenetics is any change in the phenotype, remember that's the outward appearance of your DNA, that are not due to actual physical DNA differences. So we're not talking about changing the base pairs here. We're not talking about changing the A's and the G's and the T's and the C's. We're changing other parts of the DNA that affects our traits. All right, a great example of this is from a website where they talk about these rats that lick their young, and that again was in the movie as well, but it shows the actual simulation of what those histones would look like in those rats. So you can see in this first picture, these were the baby rats that were licked a lot, so they had high nurturing. So they did not have as much methylation, and you can see how this is loosely wound DNA, right? The DNA is in gold, and it's loosely wound around those nucleosomes. And you can see a couple of those little histones sticking out of there, right? Okay. In the low nurtured baby rats, so the ones that were not licked a lot and not taken care of a lot, you can see how tightly packed those nucleosomes are. And maybe you can see those little green dots, the methyls, that are now attached to the histones. So the methylation occurred, which made the nucleosomes bind up closer to each other, wind the DNA tighter, and that affected the appearance or the transcription of the genes in those areas. So those genes are less likely to be transcribed, kind of like it was turned off. And remember, less methylation, like in this picture, means the genes are turned on. In other words, they can be accessed, they can be transcribed. Okay, now to go a little bit beyond the epigenetics control of your genome, there are other ways that your body has built in to control which genes are turned on and off. And if you remember, we talked about the central dogma of biology in the first regulation video, where we start with DNA, the DNA gets transcribed into mRNA, and then the mRNA gets translated into a protein. And each of those steps has different ways that it can be regulated. So we're going to kind of go through those fairly quickly so you have other ideas of what goes on. So the first one is happening during transcription. Now during transcription, remember we've got our double-stranded DNA, and off of that double-stranded DNA, one of the strands is going to be used as a template to make some RNA. But before that can happen, the RNA polymerase needs to bind to the DNA so that it can read the DNA as the template. Well, we have different types of molecules, usually protein molecules, that are activators and repressors. So some molecules help the polymerase to bind more easily to the DNA, and some hinder the polymerase from binding to the DNA. So essentially we can turn those genes on or off depending on if that polymerase is able to do its job, if it's able to transcribe the gene. After transcription, so here we're post-transcription, remember we talked about the fact that the RNA can be edited, it can be spliced. So if we have a stretch of RNA that's got 1, 2, 3 sections, and we're going to make number 1 an intron, and number 2 an exon, and number 3 an exon. Well, technically, there are a couple different ways that this could be spliced together. Remember, no matter what, we're going to get rid of that intron, right? They go away. But the exons may or may not be included. So version 1 is just this exon is edited, so this one is used, the other one is spliced out. Or version 2, we could have this exon used. So not all the exons are used in every version of the RNA. So there's an alternative. There are choices as to which exons actually get passed on to translation. So in other words, sometimes it's not just the introns that get spliced out, sometimes exons could be spliced out as well. So again, another way of controlling which genes get translated. Also in post-transcription, remember when we had our RNA molecule here again, and there were 2 special parts to it? We had a 5' cap, and we had a poly A tail. Well, what happens is there are enzymes that can come along and start to degrade that poly A tail, which then triggers the degradation of the 5A cap, which means the third enzyme will come in and destroy the whole mRNA. So the mRNA can be degraded or destroyed by an enzyme if a series of events happens that triggers that. So again, that's controlling the genes, right? If we want to turn that gene off, We can just destroy the mRNA and that product will never be made. Also a post-transcription, so we still have not yet gotten to translation. Again, we have our RNA, so single-stranded RNA, and we have potentially these little RNAis. They are little snippets of RNA, and I'm just going to draw them like this because I want to show you the base pairs. So it's just a little snippet of RNA. It's still a single strand, just like all RNA is. And what happens is it can come over and bind with some of the base pairs from the RNA transcript. So the one that just got transcribed, that little RNAi might come in and fit with it, binding it into what is now a double-stranded molecule. Well, that's not going to work for translation, right? When translation happens, when it binds with the ribosome's not going to be able to read a double-stranded RNA molecule. So that's one way this could control translation, is it just is not readable anymore. Also, there are enzymes that recognize this and will destroy anything that looks like this. RNAis are actually a pretty new discovery in our cells, and the reason scientists think we have them is because they believe the body evolved this way to deal with viruses. So a viral RNA came in that was single-stranded RNA, and we had this RNAi that could bind with it. It was like a tag to tell our enzymes, destroy that. That's bad RNA. So that was the original purpose of this RNAi, but it also works to regulate our own RNA that we might want destroyed because we want to turn a gene off. So pretty fascinating stuff. All right, we're getting to the end here, finally to translation. So this, remember, is when the ribosome hooks to the RNA and we read it to create a protein. So just like in the beginning of transcription, I said there were proteins that could bind to the promoter area and stop polymerase from doing its thing. Well, in translation, there are also regulatory proteins that might prevent initiation. So they prevent initiation, so they stop the binding of the ribosome. Again, potentially preventing any of those genes from being translated, so none of those proteins are being made. And then the last one, something that could happen in translation. If a cell has been or the cell's DNA has been replicated, back up. So if the mRNA, so that single strand again, remember that had the 5' cap and the poly A tail. Okay, so this could be translated multiple times. So a ribosome could come through, decode the gene, make a protein. And then another ribosome could come through, decode the gene, make more protein. So it's going to continually be read to make more and more and more and more protein over time. But as it does that, this poly A tail is going to get shorter and shorter and shorter and shorter. Okay, it degrades over time. And so what happens is there are enzymes in the cell that recognize that this poly A tail is getting shorter and basically that RNA is getting old, and they come in and they eat it up. They destroy it. They break it up. So that's one more way to regulate or control how much protein is made. So if an mRNA molecule has been transcribed enough, or translated enough, we've made enough protein, those enzymes will recognize that poly A tail is getting shorter and will destroy it. Okay, that's it for the regulation of the eukaryotic genome. Make sure that you go ahead and answer the questions that go along with this video so I have an idea if you have any areas of confusion.